To open up our program this morning, we're going to have our research update, which I know is many of you, your favorite part of the conference, to really get to know what's going on in research, where we've come from, and where we're headed, what we can look forward to in the future. And we have an all-star panel for you this morning to talk about research. So I'm going to bring up our panel. If you can, come on up. I'm going to bring up our panel that's made up of Dr. Leonard Milstone, Dr. Keith Choate, and Dr. Amy Paller. This is our all-star panel today, so you're going to get all the best information. Why don't you kick it off? I know. Yeah. Okay, great. Starting us off is Dr. Milstone.
Okay, good morning. We're all here because we want to have better treatment for our family and our friends and our patients with ichthyosis. And I'm just going to give a brief little introduction to get you thinking about the different ways in which people in the pharmaceutical industry and in the laboratories think about designing new therapies. You'll hear a lot of good details from Dr. Paller and Dr. Choate, and I just want to give you sort of an overview. So just to remind you, the reason we can even be thinking about this is that when we know what your gene is, that's where the DNA is, we then can predict what the RNA will be that makes the proteins. It's the proteins that really cause the disease, and those proteins are responsible for doing a bunch of things like making structures in the epidermis, generating metabolic pathways and signaling pathways, and all of those things eventually lead to the symptoms that all bother, that are the things that bother you, the scaliness, the redness, the itching, and those kinds of things. Oops. Okay, and when we're thinking about how to treat diseases, most of the treatments that you've all experienced are way down at the bottom there, the signs and the symptoms. And I'm not demeaning this. These are very important because these are the things that really bother you a lot. But as investigators, we want to get closer to the origin because in a monogenic disease like ichthyosis, once you know what the gene is, you can sort of predict what those other things in the pathway will be. If you don't know what the gene is, if you have psoriasis or eczema, you have to treat mostly the symptoms and hope that eventually the genes will be discovered and you can move forward. But that's different for ichthyosis right now. And so right now, we are hoping that there will be some good treatments for the structures and the metabolic pathways. I think Dr. Choate and Dr. Paller will talk to you about some of those things. Obviously, in the future, if you could replace genes that aren't working well, or the proteins that aren't working well, like the transglutaminase that causes many forms of lamellar ichthyosis, that would be wonderful, or the enzyme inhibitor, lecT, that is important in Netherton syndrome. If you could replace that in the skin, that's wonderful. And you could think about doing that by adding a protein back. You could also think about interfering with the RNA that makes those proteins if the protein is a bad protein. And that's been done. Those of you who came to the talk I gave yesterday, there's been some examples of using a small interfering RNA to interfere with the RNA that makes a bad protein. And finally, there are examples where you might actually replace a gene that doesn't produce a normally functioning protein, like people have just tried to think about adding the transglutaminase gene back, and either directly or in transplanted cells. And so those are the kinds of things that we as investigators start thinking about. We're still sort of at the bottom. We're hoping to move up, and I think there will be a few discussions about some of the more technically advanced kinds of therapy, but that's what I'm going to have to say.
Okay, get this off. Okay, so Len nicely set me up. I'll try not to overlap too much? I'm going to move over here so I can see my slides. I'm going to take you on a bit of a tour, and some of these slides will look a little complicated, but I'll just try to say things in a simple way to walk you through at least where I see the research going right now in some new directions in a lot of different angles. We're going to hear from Keith, too, with more research after this, but I'm going to start with some simple ones. We're going to talk about some things that are pie in the sky, and certainly some that are ongoing or about to move forward. So I just want to make a simple recommendation to start with, and we've had a little discussion about this at this meeting, and that's that I don't really know fully the role of vitamin D. Maybe you do more. Dr. Milstone spent a lot of years looking at vitamin D and parathyroid-like substances in skin, but we certainly know that vitamin D is important for our immune system and that it increases the natural antimicrobial, antibacterial proteins in skin in response to when there are organisms that shouldn't be there. We also know that rickets, which is from profound vitamin D deficiency, has been reported in several children with ichthyosis, and I know I've seen it myself. So I just wanted to show this was a study of 87 patients with different forms of ichthyosis. Now, you might recognize yours by the abbreviations at the bottom. And almost uniformly, there was a deficiency. Under 30 is deficient, under 10 is profoundly deficient. And these are means, so some people weren't deficient, but certainly the means were all below, and a lot of these individuals were quite deficient. So I just mention it because it's so easy to get that checked. It's so easy to take some vitamin D supplementation. We prefer to take it orally versus sitting out in the sun. But as you know, ultraviolet light also has some positive effects. A little bit more about vitamin D. I will tell you that there is a physician scientist who's very interested in ichthyosis who was in India when he did these studies. And he took several patients who had autosomal recessive congenital ichthyosis or epidermolytic ichthyosis who were quite deficient in vitamin D. And he supplemented them with very high doses, so 60,000 international units per day. Disclaimer, do not do this on your own. That's a very, very high dose. And if you think about taking vitamin D beyond a few thousand units, you got to talk to your doctor. But at any rate, this is treatment for severe vitamin D deficiency. He gave it for 60,000 international units per day for 10 days, and then he went on a standard kind of dose or a little bit lower, depending on what age they were, 400 to 600 international units per day. Marked improvement, not just in their rickets and their vitamin D, but their skin dramatically improved. It hasn't been a whole lot since that time. I think there's been some work on it, but I did pull this recent article that just came out this year with 24 patients, mostly with autosomal recessive congenital ichthyosis, who completed a study of just 2,000 international units a day. And this was called a double dummy study where everybody got a placebo for one and the active for the other, and they were randomly assigned to either get acitretin, the retinoid, or to get vitamin D. And although the retinoid did look like it was a little bit better here, I don't think the vitamin D did too badly in this study. There was a significant reduction at the first visit checkup and the second with quality of life, and at least from the beginning to the next one, there was a significant reduction as well in the severity score. So just something that's just so easy that you ought to think about working with your doctor to try it. And obviously, if you want to do it right, get a level first before you do this. A little bit about retinoids. There's a wonderful article that recently came out, and many of the authors are the physicians who've been at this meeting that look at the use of retinoids for ichthyosis. And so I just listing it here. It was a wonderful review. It went over who benefits from it, counseling about potential risks, tips about giving retinoids to children. So I just say, does your doctor know about this? And you can jot it down if you don't, or mention to him. It was in pediatric dermatology fairly recently. And we were talking yesterday about maybe seeing if we could get the entire article posted on the FIRST website. I think we've heard about topical retinoid, topical isotretinoin. Oral isotretinoin is used a lot for bad acne. and is also used for many patients with ichthyosis. It's one of the 2 retinoids we have here in the United States to be given orally. This is a topical formulation of it that has not been available. And the early studies, I'm just showing you some pictures here, looked quite good. And the next phase, phase 3, is currently now moving forward. Now, you heard about this from Leonard Milstone a minute ago. I'm just going to remind you, it is genes that we think about if we're thinking about cure, because we have to change the gene to be able to do something that's permanently going to effect a change. That's going to be tough, and it's not going to be in the next 5 years easily, maybe not even the next 10 years. It's something we have to keep trying to do. There are studies now in other diseases, particularly in a blistering set of genetic diseases, where you take skin cells, you put the gene in that's the full-length, perfectly normal gene when something is missing otherwise because you don't have a normal set of genes, and you then expand those skin cells and graft. And that's easier to do when you have a blistering problem that takes away the skin. Here, you'd have to probably take off the skin and put that on and replace large areas if we're talking about ichthyosis. But that work is moving forward, particularly in Europe, with both Netherton syndrome and also starting to be looked at for lamellar ichthyosis. Gene introduction, I just want to mention, also with the blistering set of disorders called epidermolysis bullosa, there's been some very interesting work done with gene introduction, but not in such a way that it permanently incorporates. So it basically gets into the cell and turns our cells into a factory for making the protein that's missing. And this is being done quite successfully with a blistering problem where there's no skin barrier and you can easily get it into cells. We're not sure how well it would work for something where there is at least some barrier, although we know it's not great in ichthyosis. But this is something that there were some early studies with. You've probably heard something about this, but I do want to tell you, I don't think this is going to move forward, and I don't know that it would be practical if it did. This is going to be very expensive therapy, and the idea of putting it over every part of the body, which anybody with ichthyosis would want to be able to not just treat the face or not just treat the hands, but treat a large surface area. I think that's going to be something very difficult, and I don't know that this company that was thinking about doing it for others too, like Harlequin and maybe other forms, may not be pursuing this, but we'll keep you posted on that. So what can we do now? And we heard beautifully from Dr. Milstone about this whole concept of what's downstream of the genes. And that's where we're looking right now to target while studies continue to improve on the idea of curing through genetic change. So reminding you, DNA to RNA to the protein, which is actually what functions in our cells. And so we can look not at the DNA, but also at the RNA and see what kind of changes there are to get an idea of what's up, what's down, what there's too much of, what there's too little of, and then understand how that may be leading to inflammation, scaling, itch, pain, etc. And we just published a study that I want to thank so many of you for because it involved biopsies that we've done, including at this meeting, where we looked at 56 patients, and I'll show you that in a minute, and really looked at the RNA changes across the spectrum of ichthyosis. One can look at protein, and there are some ways that we can do this. In fact, with the tape strips like we were doing at this meeting that so many of you offered to participate in, including techniques like O-Link, where you have from just 2 of those tape strips enough to be able to look at a large panel of proteins. And then there are other ways that do require biopsies. And lipidomics. Remember that we have proteins and lipids in our barrier, and the lipid changes can also be looked at in large numbers, either in something called targeted or untargeted. And we just completed a study with 77 individuals with ichthyosis using the tape strips that we're putting together now. Now, what about this concept of not treating at the gene level? Can it work? And I just quickly want to show you, none of you in this room have a family member with Child syndrome that I know of, but this is a form of ichthyosis, and we've known for a long time that it's due to having an enzyme that's missing in a pathway that leads to cholesterol. Cholesterol is very important in our skin, and in this case, knowing that, the thought was, can you replace what's deficient because of having a problem with a pathway? But when we topically added cholesterol, and I have to give credit to one of the grandfathers here at first, Peter Elias, who really has done so much of this work with these lipid pathways, and he and I together thought about this. We added cholesterol, it didn't do the job. But it so happens that we know that at the very beginning of the pathway that leads to cholesterol is another enzyme that is rate limiting and can be inhibited by something called statins. I think many of you may have heard of statins, and maybe many of you are on statins. And so we were able to topically compound something that's now used around the world for this rare form of ichthyosis that included the statin and included the cholesterol together, and was able to prevent the buildup of the toxic in-between components of this lipid pathway that were causing toxicity to the cells. Now, it's probably not as important now, we know, to have the cholesterol as to have the statin blockade, but it's something to think about, just showing you in this girl how rapidly her skin became normal. And this has now been leveraged through work that Dr. Choate has done, and we've worked on this together, for another set of disorders, all of which also have their genes in this pathway called the porokeratosis, another disorder of cornification, like the ichthyoses. So I'm showing you this pathway. This is something that Dr. Jason Meyer here is working on with congenital ichthyosiform erythroderma, and many of you have this. And look carefully at this diagram because you might spot those letters and numbers that make up the name for the gene that you know is altered for your family. And these are a series of genes that produce proteins that are critical for the production of some very unique and very long ceramide lipids in our skin that are critical to the formation of the barrier. And when you don't have one of these enzymes, when you block that pathway from proceeding, you don't have the end product of these very special omega hydroxyceramides that make our skin barrier. Now, we might be able to bypass this whole thing and treat everybody with CIE by putting on that missing lipid. And that's the goal. But I would also say, thinking about what we learned from child syndrome, we really have to think about whether there's any toxicity at different steps of the way as well, to the buildup of what comes before. Now, this concept of blocking something that's downstream is also moving forward for Netherton syndrome. I know we don't have very many here with that, so I'm going to just quickly mention that this is a process where there's a deficiency of a protein that inhibits some proteases that, when they're increased, can break down skin. So they break down the skin barrier, but they also drive skin inflammation. And the idea here is that if we can inhibit these kallikreins that have increased activity by either topically applying or giving systemic kallikrein inhibitors, or even doing something that adds the inhibitor of the kallikreins that's the natural inhibitor called Letke, we may be able to achieve the goal that we would achieve by gene therapy. In the meantime, we've got lots of different forms of ichthyosis, and I'm going to just quickly remind you some of the work that we did and just update you on where that is right now. So we knew that all these forms of ichthyosis had scaling and thickening of skin, redness, and wanted to know, with what we were seeing coming out with some of the common disorders, whether there might be a pathway that resembled a common disorder for which we could leverage some of that therapy towards this. So we did studies that looked at the RNA. not the DNA, not the protein, the in-between RNA, what goes up, what goes down. And you'll see these, what are called heat maps, where red means it goes up, blue means it goes down, white means something in between. And the first row here shows, for example, things that are not produced too much in blue in the healthy skin because they are, they're just not produced as much compared to what you see here in the various columns. And the first column you see after the healthy skin is psoriasis, a common inflammatory disorder with redness, scaling, and thickening. The next one is eczema, and the next one is a whole group of skin, in this case biopsied skin, from ichthyosis. And you can see that there's a lot of red. There's a lot that's going up, and it looks very much like the pattern that we see here for psoriasis. And in fact, what are called Venn diagrams on the left side show where there's the overlap. And there's lots of numbers there that are overlapped in red, numbers that are overlapped in blue going down, that is overlapping with psoriasis and not eczema. And this has been shown by a variety of tests that look at the RNA. This is just simply showing that, in fact, the increases and decreases correlated with the severity of the ichthyosis, and particularly the severity of the redness in the ichthyosis. And a test called RNA-seq, which was what I was talking about, just came out with 56 individuals with congenital forms of ichthyosis, showed the same kind of thing with more red compared to the first column there that's healthy. And it turns out that most of these are genes that are in a pathway that is involved in a certain kind of inflammation, where something that goes up is called the interleukin-23 Th17 pathway. It doesn't matter the name, it matters the fact that we know a lot about this because it's the same pathway in psoriasis. Now, it's also the pathway that increases in response to organisms on our skin. And we know that if you have ichthyosis, you have an increased risk of having particularly Staph aureus bacterial infections and also perhaps fungal infections. So one thing we did that is really being moved forward even more extensively by Dr. Milstone and his group is to look at what's called the microbiome of skin. And we ended up doing this in 22 individuals with ichthyosis in some studies that we were doing, and that's recently been published as well. And we found an increase in Staph aureus, and we know that Staph aureus in itself produces proteases that break down the skin barrier and cause our own cells to produce more proteases, and it also stimulates the immune response. We saw that there was an increase not just in Staph aureus, but also in a lot of the good staph that can be seen, and that's a response that our bodies have when we have too much Staph aureus in an attempt to try to fight that. We also saw an increase in another bacterium that also we know goes up to control Staph aureus. We saw a decrease in the diversity of the bacteria. That's the number of different bacteria, and we're largely talking about good bacteria here. And we saw a decrease, not surprisingly, in certain bacteria and also in a fungus that's normally on our skin, but really requires lipids of different kinds to grow and do well. So it wasn't surprising with the dry skin of ichthyosis to see a decrease. So we know that that increase that we found might just be related to that, but across the board in ichthyosis or in certain subtypes, could there be a role for what we found in terms of that immune change that resembles psoriasis? And if that's the case, then could we, what's called repurpose, could we take something that's on the market for psoriasis and try it for our patients? I do want to mention also that this work allowed us to look in the blood, and we talked about this in the joint session the other day, because we found evidence that ichthyosis is not just a skin problem, but actually there's evidence of activation of the immune system internally as well. That's called systemic immune activation. And don't look at the words here, they're very complicated, don't look at the graph. But I just wanted to show for those of you who want to see the actual data, that when we're able to take cells in blood and sort them into the different cell types, what we find is that there are increases in the numbers of cells that, one, show a marker for activation, and in fact, that marker for activation was higher in ichthyosis than in eczema, psoriasis, other disorders that we know have immune activation systemically. And also we found that the type of cells and their expression patterns are the same type that we see increased in psoriasis, where we know that there are certain systemic components. We also found increases in the serum itself of some of these circulating activating types of proteins. So that raised the question, do we see some of the same changes that we might see in psoriasis? So psoriasis can be associated with arthritis. Psoriasis can be associated with some cardiometabolic problems. And so we're starting to look at that and really need to pay attention to the fact that joint issues may come on earlier. There may be more, and understand them better and see whether there's anything to this. But I would say that if we start to find these internal components in systemic immune activation, it raises the question of whether we should consider being more aggressive. Maybe there's a reason to give something systemically rather than topically, especially if there's safety associated with it. So what about the repurposing? I'll just take a quick few minutes to go through that and where we are. I'm just showing you here what is out there for psoriasis. The inhibitors, IL-23 or the IL-17 pathway, that are available, and these are all available in blue for adults. The ones that have red around them have been approved for children 6 years of age and above. I'm also just going to mention, because I know somebody sent in through the HOVA app a question about dupilumab. That's off on the right here. That's a medicine used for eczema, and I'll mention it briefly. I don't talk about brand names, but they are at the bottom of the ones that have been used most commonly to try to look at ichthyosis. So anecdotal reports have been coming out since the discovery of this shift towards the psoriasis-like picture, particularly with Netherton syndrome and CIE, and I'm showing you some of them here. So I'm showing pretty dramatic changes. So I think the question is always, we pick and choose what we put in the literature when we publish it. We publish the good stuff. We don't publish the stuff that doesn't work. We should publish the stuff that doesn't work, but we don't. So one of the questions is, are these just a few responders being reported, which I think is still valuable. You know, I mean, anybody, even one person who responds to something, if it's a true response. So we did an exploratory, two-centered, double-blind, randomized, placebo-controlled 16-week trial, meaning we had two groups. We did this across the spectrum of ichthyosis. One group in the first 16 weeks got placebo. The other group in the first 16 weeks got one of these inhibitors. It's called secukinumab. And we looked at how they did. And we looked at the safety. Was there an increased risk of infections, particularly knowing the baseline risk, and how well it worked. And I'll show you the data here. You can see that after the first 16 weeks, everybody went on the active medicine, and we really paid attention to what happened over the next 16 weeks, particularly the ones who were originally not on it, and then they could continue for the full year on this medicine. So, first of all, a co-primary endpoint was safety, and we saw no safety signals, so that was a good thing. And we had some patients who did really well. I'm just showing you some here. At the top is somebody with Netherton syndrome. We have a few who did well with that. And the bottom is a type of congenital ichthyosiform erythroderma that's related to variants in NEPAL4. I know some of you do have that subtype. But I want to call your attention, it's a little bit cut off, but what's on the left is a whole series of what we did, looking at severity, looking at quality of life, looking at itch, looking at pain. We tried to be very comprehensive. And on the right are the p-values. And if you don't know anything about p-values, what you want to see is a number that's less than 0.05. And none of these had a number that was less than 0.05. So that was disappointing and showed us that across the spectrum of the ichthyoses, this is not going to be the answer. It won't work for everybody. But we were encouraged that several of our patients did well and wanted to stay on the medicine. And I think that what we need to figure out is which ones. Well, I will tell you that nobody with lamellar ichthyosis, at least in our study, did well with it. It just didn't seem to work for that. Several people liked the fact that they had to put on less moisturizer. They thought they looked better with epidermolytic ichthyosis, but I wasn't impressed when I looked. So I'm not excited about it for that. But I do think that several of our patients with congenital ichthyosis from erythroderma and Netherton syndrome did do well and deserve further study. But I also want to mention the importance of double-blind, randomized trials like this. And I love having open-label studies where everybody gets the active. That's what we all want. We want to be in a study where we know we're going to get something that at least could work. But the problem is that the world doesn't turn like that, and you can't know. In this study, there were people who were on the placebo who did better in that first 16 weeks. Ichthyosis, as you know, and particularly certain kinds of ichthyosis, you have good days, you have bad days. If you happen to come in for your evaluation on a day when you're having a good day, it's going to look like you're improving, and we're going to give you a different kind of score. So I just want to mention that, first of all, I want to encourage everybody to take part in trials. You have to make some sacrifices, at least for a few months, to do these kinds of trials. But the end point, if we can discover, and we're not going to discover without doing these kinds of trials, could be very valuable for you and for everybody in this room. So be willing to be in trials, but understand why we have to do these double-blind trials. Okay, so what about dupilumab? That was a question that was asked. It targets a different pathway. It targets a different shift in the immune system. And I will say there have been some responses with Netherton syndrome, but there also have been many that didn't respond. I've had patients who did not respond with Netherton syndrome to this. I haven't seen anyone yet who responded in terms of their skin getting better, but some reports are out there. And there's also, this was a report, for example, where the patient did well in the first 6 weeks, and then at 8 weeks, you can see how the skin looked. And so, again, tremendous variability with Netherton syndrome in particular. What I am interested in is that dupilumab has been shown to have an effect on itch through dampening the nerve responses to all sorts of itch stimuli. And so we're now doing a study trying to look at how dupilumab might be affecting itch, but it's not doing a lot to anything else, and we'll see where that goes. Others are looking at that as well. So the bottom line is, what are the predictors here? Does age make a difference? I don't know how much it does, but a lot of the patients who were reported anecdotally were younger. So an ongoing trial right now of ustekinumab that we're doing in children, we're seeing the greatest responses with Netherton syndrome and CIE Nepal-4, just like what we've seen in the literature and in the previous study. We do have 6 children with harlequin ichthyosis who are in the study, and I will tell you, I'm not seeing dramatic changes in how they look, but there is a reduced burden reported, less moisturizer required, just feeling better in general, so we'll see where that goes. Hopefully biomarkers will be helpful in understanding how to predict. We didn't really see biomarkers helping terribly much in the secukinumab trial that we did, but we just looked at certain ones, and the advantage of big data, like proteomics or transcriptomics or lipidomics, is that we can get back thousands of data points, and it might just be something that we don't even think to look at right now that is a predictor. So I'm hoping with lots more in trials, time that we can find these predictors. Now, I do want to make a plea to you. With this work, we've put a lot of time into it. I know there are people who've come up to me at this meeting and have said, I'm on this inhibitor, and look at the difference. It has truly changed my life, including some forms that aren't talked about here. We know that there are some rare forms as well that seem to universally help patients, like erythrokeratoderma with cardiomyopathy, or another type called SAM syndrome. And so we're trying to put this all together. We have an international group with the European Research Network, with the Pediatric Dermatology Research Network, that we've been surveying to find out anyone who has tried one of these biologics. I want to know if it didn't work. I want to know if it worked, but I want to know if it didn't work, so we can really start to understand what's going on here. Is it worth recommending these, and for whom? So my email's up there. Anybody can send me an email at any time, but also in particular, if you've been using one of these, I'd love to talk with you to fill out a form so we can include your response. Finally, I just want to remind a little bit about the tape strips that we did at this meeting. It's amazing. We can actually get information from something so painless and non-invasive. So I want to thank the almost 70 people who donated their arms, and some flakes of skin to us at this meeting, and we hope that we're going to be able to use this in the future, and that other scientists will be able to use what we collected today to ask questions. Finally, I do want to say that it's not just trying to figure out treatment responses, but understanding why epidermolytic ichthyosis looks different from CIE, looks different from lamellar ichthyosis, looks different from Netherton syndrome. How we as doctors look at the different clinical pictures and say, you have so-and-so. And we know because of the way the scale looks or the way the redness looks or the distribution, but we don't know on a molecular level what is between the genetic change and what we see on the skin. And these kinds of technologies are going to help not just with hopefully being able to predict responses to medications, but also helping us to understand. And this is just showing you from the article we just put out that if you look at, again, this is called a kind of a Venn diagram where we have areas that overlap are the same and areas that are different. And I just point out in the boxes the big differences that are unique to each of these forms of ichthyosis shown here, and we hope to be able to learn from them. And we have other types of analyses too, metabolomics, where we look at the metabolic changes, epigenomics, where we can look at environmental factors and how they regulate what genes are expressed. So lots of work to come. Very quickly, if you're interested in clinical trials, we're going to hear about this after this in the next session. Free, clinicaltrials.gov. You can go on and check out what's going on with ichthyosis. It's really easy to see. And again, I remind you that new products are only becoming available if you participate in trials. There was a recent trial. I know I heard from at least one individual who was very upset about this, but it had to be discontinued because there just weren't enough people with a promising new approach for ichthyosis. So be generous. Quickly, questionnaire studies. We've had some at this meeting. Thank you for participating. We hope to be able to give you information about transitioning to adulthood and taking care of ownership of disease, knowledge about ichthyosis, and bullying. And bullying is a big problem. I will tell you that in the Pediatric Dermatology Research Alliance, the big study that was done in the last few years looked at stigma across a variety of particularly more visible skin problems in children, and ichthyosis, and of course several of these others, truly have stigma that's at the same level as what we see with neurologic disorders, cancer, and we've got data now about the effect on anxiety, depression, peer relationships that we're going to be able to show to the FDA, to industry, to others who might not have thought about the issues related to skin disease and how significant they are. I also want to encourage you, if you didn't take a picture, take out your phone and take a picture here of the QR code. I want to give credit to Andrea Rusted, who's in the back, a medical student who's been interested in being part of FIRST and involved in ichthyosis for the last few years in her training, and I'm sure will be a pediatric dermatologist coming to this meeting for years to come. And she became interested in bullying from talking to many of the children in the Chicago area, and decided it was really important to write this book. And I encourage you to check it out online. But I also finally want to end by thanking the many who sent in pictures. Some of them were out of focus, but we tried to put in anything that was in focus. And so in this book, there are many of you as well as tips. And finally, I want to thank FIRST, because FIRST helped to fund getting this book out as well. So thanks, all of you.
So we're slightly over time, and so what I won't do is talk about the discoveries from my lab, but instead talk about the registry. Because I think the registry is really probably one of the most important things that FIRST can invest in now to be able to be ready for when clinical trials are available that actually will have efficacy across multiple different skin types. And so while I'm primarily a human geneticist who identifies new genetic causes of ichthyosis so that we might be able to comprehensively understand the pathways that rule them all, so that we might be able to come up with single molecule agents to treat multiple types of ichthyosis, I'll focus on the registry itself. So the registry has a lot of information. For those of you who've been a part of it, you know that we collect information about where you live, who your family members are. We have you fill out a big questionnaire that tells us a lot about your birth history, about your medical history. We get information about quality of life. And ultimately, we collect gene-level mutation data. So I want to make it clear that the registry is not only for people who don't have a genetic diagnosis, it is for everyone, because the purpose of the registry is to connect families with research projects that are relevant to their disease type. We're integrated to be able to do surveys dynamically. We consent folks to be notified about future research, and in fact, many of you in the room have been notified via the registry about specific studies, and it provides an incredible resource. We now have a genotyped, that is, we know the gene, phenotyped, we know what the disease looks like, a group of people who are ready to participate in research. And so some of you have been able to enroll in meetings where we can do things perfectly, and we have photographers on site, and we can collect saliva for you, but we also have the capacity to do a lot of this by mail, because everyone has a great smartphone, and so we put together a guide to enrollment that allows people to take photographs that are representative of their skin so that we get a sense of what their skin looks like, and we can also collect DNA via saliva and have you send it back by mail. This allowed us to really expand our enrollment. And so we now have 1,600 families that have enrolled in the registry. We've provided genetic diagnoses to 1,300 of them, both in the United States and worldwide. We do this by pre-screening a number of genes initially, identifying mutations in 85% of subjects, but then use exome and genome sequencing to identify rare causes of ichthyosis and many new genes that cause ichthyosis. And we currently are working on 4 new causes of ichthyosis that have not been previously reported. Some, we're very happy to partner with some of the folks who are here today in that work. And so in the interest of time, I'll move on to a very important report that came out of the registry via Mary Sun, Teddy Zaki, and Nurey Maroukian, who many of you met. And this is a paper which describes the genotypic and phenotypic landscape of ichthyosis in 1,000 kindreds, many of whom belong to FIRST. And this educated providers, and we chose not to publish this in a technical scientific journal, but rather in a dermatology journal, so that we could show providers what the spectrum of phenotypes is for mutations in individual genes. And I've met many of you who say, I have ABCA12 mutations, but I don't have classical harlequin ichthyosis. And so we wanted to show the spectrum from essentially normal skin to mildly affected, moderately, and severely affected, all within one genotype. We also wanted to show that people can get completely different presentations than you'd expect. We were able to identify 266 novel pathogenic variants in 32 genes. This means that instead of getting a report that says that you have a variant of unknown significance, when you get your genetic report, you're actually going to get useful data, because we now are able to rapidly move to causal mutations. I think this has really transformed ichthyosis diagnostics clinically, and also expands the phenotypes due to mutations in known genes. And so we've been able to develop a lot of clinical tools from the registry. We've been talking a lot about trials. One of the most important things about trials is being able to prove that the drug works. And in fact, many trials that have been initiated in ichthyosis have failed because they haven't had good measures. And so our group with Dr. Milstone for the VIS, with Dr. Paller and many of the members of the MSAB for the ISS, and many of you actually participated at this meeting in an in-person validation of the ISS, we're able to create detailed assessments of clinically relevant sites that are valid both with photographs and in-person validation. Timber used the VIS very successfully in their phase 2. They're using it again in their phase 3. But I think the ISS will really be the instrument that helps to rule the day and can give us information about most subtypes of ichthyosis. The registry is working hard for you to enable trials to be fully enrolled. For rare diseases like Sjogren-Larsen, we've worked with investigators interested in basic questions like Dr. Milstone, Timber, Maine, Anapsys, and countless other companies. And we've also been facilitating basic science. We are providing cell lines to people who are interested in new genomic technologies. And so when people give us a biopsy and consent to have us transfer that to other investigators, we've been able to provide these cell lines to make things possible, like the Crystal HSV-1 gene therapy study, individuals who are looking at CRISPR, and numerous pharma companies that are interested in other subtypes of ichthyosis. So there are new clinical studies that are being initiated by the registry now, and I'm going to ask Caroline Echandia-Francis, who led the clinical screening program, to talk about growth and development. This is a study which you should all be a part of because it will answer one of the fundamental questions about what is the natural history of ichthyosis in young people, and how much support do we need to provide to early families early in life.
Hi, I'm Caroline. I just completed my first year of medical school, and I've been working with Dr. Choate for about a year now. I'm going to be talking about growth and development among children with ichthyosis, which is a study that was recently launched in early 2022. You guys have probably seen the flyers hanging around the hotel that have the QR code, and they were on the tables previously. So thank you to those of you who have already participated, and I hope that this presentation excites the rest of you to participate as well. Okay, so what is the study investigating? We were eager to do this study because there have been long-standing observations by physicians that children with ichthyosis tend to be smaller in stature compared to their unaffected peers. And there have been previous studies that have looked into this relationship between growth and ichthyosis, but these studies tend to be smaller. So most of them have about 12 participants or fewer. And to date, there is no large-scale study that looks into the relationship between growth and ichthyosis. And this is the knowledge gap that we are trying to fill. When looking at previous research as well as physician observations, the hypothesis that we ended up forming was that certain types of ichthyosis specifically, such as Harlequin or X-linked, for example, are maybe more closely related to growth delay in children. And so we don't know the results of this study yet, but hypothetically, if we do end up learning that there are certain genotypes of ichthyosis that are associated with growth delay, what we are hoping to be the goal of this study is that parents and physicians can say, okay, we have a newborn child who was born with Harlequin. We know from this study that this child may experience delayed growth, and that the averages of growth delay are this amount. And so are there potential early life interventions that can be implemented to close this gap between this child's growth with Harlequin and the growth in unaffected peers? So who can participate? Well, we're inviting all people with ichthyosis who are under 30 years old to participate, and we're also interested in recruiting their unaffected siblings. This 30-year-old cutoff mark is just because physicians don't tend to keep pediatric records forever, so if you stop seeing your pediatrician at 18, they're probably not going to have them when you're 50, so we set the cutoff at 30 years old. Participation takes about 5 to 10 minutes. What you'll need is your physician's name as well as a way to contact them. So their fax number, their phone number, their address, or a combination of those three. And you'll fill out a survey, and what it does is it gives us access to your pediatric growth records. So we'll be looking at metrics such as your height, your weight, changes in head circumference throughout those pediatric years. And we'll compare those numbers to the averages of children of matched to age and sex as we move along your growth chart that are provided by the World Health Organization. And then we also want to do the sibling comparison and compare your data to your unaffected sibling if you have one. And that's because siblings share more of their genes compared to strangers. So it increased our confidence that what we're seeing, any differences in growth, are in fact due to the gene mutation that is causing ichthyosis and that gene's downstream effects on growth. So it basically is going to give us more confidence in the findings that we get from the study. Okay, and here's the QR code. This can be scanned with your phone, and that will encourage you to, it will allow you to participate in the study. And then we also have here the Twitter and the Facebook that has links to the study as well. And I post the flyer, like a digital form of it there too, so it has the QR code so you can easily participate. It has a bit more information on what the study is looking at. And yeah, that is all. So thank you guys so much for your time and attention. It's been an honor getting to know so many of you this weekend. And I appreciate being invited to this and being able to get to know so many of you. I'll pass it back to you.